Hi, all of you. So, this session is a continuation of the previous session on the biochemistry of fat state. Okay, so now we'll see about biochemistry of fasting state, right? So, fasting state, the main hormone in the fasting state is the glucagon, right? What is the main hormone? It is the glucagon, and there are four phases. Yes, four phases in which the first phase is early fasting state. So we all know which is a fed state. It is about 2 to 4 hours of food intake, right? So early fasting state is about 4 to 16 hours after food intake. Next, the fasting state is about early fasting is 4 to 16 hours post food intake, and fasting state is about 16 to 48 hours after food intake, and prolonged fasting, otherwise called starvation phase, is about 2 to 5 days after food intake. And prolonged starvation is not eating food for more than 5 days, right? So these are the four phases of fasting state. Now the pathways which are active in the fasting state. So in the well-fed state, we have adequate amount of nutrients, your glucose, amino acids, and fat. But here in fasting state, we do not have adequate amount of nutrients, where glucose is a major fuel. So the first thing which we would try to replenish is by giving glucose, by regulating the blood glucose level. So we have also in the previous session we had seen that liver glycogen is responsible for regulating the blood glucose level in the fasting state. So the first pathway which is going to be active in the early fasting state is nothing but the liver glycogenolysis. Yes, liver glycogenolysis. So breakdown of liver glycogen gives us glucose, blood glucose. All right. The next phase is about, what is the time period in early fasting state? It is from 4 to 16 hours after food intake, right? So during this period, our liver glycogenolysis is going to be active. Now the fasting state is about 16 to 48 hours post food intake. So in which case the gluconeogenesis pathway is going to be active. It's not that just it will start at 16th hour only. It will start from the 12th hour itself. So simultaneously from the 12 hours post food intake, glycogenolysis and gluconeogenesis will be starting to provide glucose to the body. All right. So in about 48 hours, it can tolerate. It can start using only the gluconeogenesis to produce glucose. Now, during the prolonged fasting, when it happens to be like you don't eat for 2 to 5 days, it's called prolonged fasting. In which case, there is excess of fatty acid oxidation, right? So excess of fatty acid oxidation will give us more of acetyl CoA. The acetyl CoA, more of acetyl CoA, instead of getting into the TCA cycle, it will be diverted towards production of ketone bodies, which can now be used as a fuel. All right. So in gluconeogenesis, when gluconeogenesis pathway is active in the fasting state, the ATPs which are necessary for gluconeogenesis to happen, there are about 11 ATPs which will be consumed by gluconeogenesis pathway. So many amount of ATPs will be provided by the fatty acid oxidation only, right? So in the prolonged fasting state, what happens? The one of the precursor for gluconeogenesis is what? Yes, one of the precursor for gluconeogenesis is what? It is nothing but your oxaloacetate, right? So first, what is the first starting material for gluconeogenesis? It's pyruvate getting converted to oxaloacetate, right? Oxaloacetate is also a intermediate of TCA cycle. It's an intermediate of TCA cycle. Now, when my fatty acid is getting oxidized, it will produce acetyl CoA. Is it not? Fatty acid oxidation gives us acetyl CoA. Now, acetyl CoA, any acetyl CoA has to enter into TCA cycle, after which it can enter into the electron transport chain and give us energy. But here, since in the TCA cycle, one of the intermediates of TCA cycle, which is oxaloacetate, is used up in the gluconeogenesis pathway, there is less amount of oxaloacetate to carry on the TCA cycle, right? So what happens instead? The acetyl CoA, instead of getting into TCA cycle, will be diverted towards the ketone body synthesis. Yes, ketone body synthesis. So this is what happens. because of excess of gluconeogenesis, the acetyl-CoA, which is formed from the fatty acid oxidation, cannot enter into TCA cycle because of the lack of oxaloacetate, which has been used up in the gluconeogenesis pathway. Alright, so the acetyl-CoA enters into the ketone body synthesis. So ketone bodies are synthesized in large amount in prolonged fasting. The next phase is prolonged starvation, in which case ultimately your fatty acids oxidation is getting over, fatty acids are completely over, your gluconeogenesis pathway is exhausted. So finally what happens, your muscle proteins, okay, your muscle proteins will start providing the glucogenic amino acids, and it will try to produce glucose out of gluconeogenesis, right? So muscle proteins are going to get degraded, and even after that, if you're not going to eat, then it will ultimately lead to death. So what are the four different phases? Early fasting, in which case liver glycogenolysis pathway is active. Fasting state, in which gluconeogenesis pathway is active. Prolonged fasting, in which case fatty acid oxidation, ketone body synthesis are active. Then prolonged starvation phase, muscle protein degradation is going to be active. It's not that just one pathway in one period it is going to be active. It will, the predominant pathway which will be active is what I have mentioned. All right. So even here, let's start talking about different organs. What happens to the metabolic pathways in different organs in fasting state, right? So first we'll start off with liver. Yes, we'll start off with liver. Why? Because in the early fasting state, your glycogen is going to get broken down and gives this blood glucose, is it not? So your glycogen, yes, the liver glycogen is broken down and gives us glucose, and glucose enters the circulation, right? The next one is gluconeogenesis pathway, which is going to be active. So in which case, what are the precursors of gluconeogenesis? Your glucogenic amino acids, your lactate, your glycerol, your propenyl-CoA, right? So these things, your amino acids or lactate is going to give either pyruvate. It will be converted to either pyruvate, or it will be converted to any of the intermediates of TCA cycle, whereby it enters the gluconeogenesis pathway and gives us glucose, which will enter into the circulation. All right. Now, what happens to fatty acids? Fatty acids again will enter into the liver. It will undergo oxidation. It will give us acetyl-CoA. Now, acetyl-CoA, as it gives acetyl-CoA enters TCA cycle, gives ATP. That ATP is used up in the gluconeogenesis pathway. But when the oxaloacetate sources become less, then acetyl-CoA is diverted towards ketone body synthesis, and ketone bodies are synthesized out of the liver. But liver cannot utilize ketone bodies, is it not? So this is about the carbohydrate metabolism, lipid metabolism, and amino acid metabolism. How is it going to help in maintaining blood glucose level during fasting state? All these happens in liver. All right. The next organ which we are going to see is about the adipose tissue in the fasting state. So what happens to adipose tissue? What is being stored in adipose tissue? Triacylglycerol is stored in adipose tissue. Right. Now the triacylglycerol is broken down to fatty acids and glycerol. So fatty acids, of course, it will go out of the adipose tissue. It will enter into the blood, where in different organs it is undergoing fatty acid oxidation, right? And then glycerol enters. Glycerol is one of the precursor of gluconeogenesis, so it will enter into the gluconeogenesis pathway, right? One is fatty acid oxidation and glycerol in gluconeogenesis pathway, right? So these are the two main organs. So just starting from the breakdown of triacylglycerol. Yes, mobilization, breakdown of triacylglycerol from the adipocyte. Right. So yes, so this fatty acid oxidation can also give us acetyl-CoA, which can enter into TCA cycle. Okay, this can happen in the early stage. Late stage, it will only help in gluconeogenesis and fatty acid oxidation. The next one is skeletal muscle in the fasting state. Skeletal muscle in the fasting state, what can happen? The ketone bodies which are being synthesized from the liver will be utilized by the muscle, right? And the muscle can also utilize fatty acids. Fatty acid oxidation can give them energy. And excess of acetyl-CoA excess of acetyl-CoA, acetyl-CoA can enter into the TCA cycle. It can be used up for providing energy. After which ketone bodies, if it is synthesized, ketone bodies are used in the formation, used in the energy giving process. Then proteins in the in the prolonged starvation phase, what can happen? The proteins are broken down. So amino acids will act as glucogenic precursors, and they will help in the regulation of blood glucose. Right. So this is about skeletal muscle in the fasting state. The next one is brain in the fasting state. So what happens to the brain? Brain always tries to use only glucose. Yes, always try to use only glucose even during the starvation phase. But ultimately when there is devoid of glucose, then ketone bodies, it can try to use ketone bodies. Okay. Now this is about the brain and fasting state. And one more organ, which is the kidneys. Kidneys in long-term fasting. Kidneys are used in long-term fasting. How was it used? Branched chain amino acids. Yes, branched chain amino acids give us glutamine. Glutamine gets converted to glutamate. Glutamate gets converted to alpha ketoglutarate by glutamate dehydrogenase. What is this alpha ketoglutarate? It is an intermediate of TCA cycle. Yes, it is an intermediate of TCA cycle. So anything which is an intermediate of TCA cycle can enter into the gluconeogenesis pathway. Right? So as a in a long-term fasting, kidneys can even help in gluconeogenesis and production of glucose. All right, so these are the different organs. What are the different organs that we saw? Liver, adipose tissue, skeletal muscle, and brain and kidneys. Right. So let's see an integration of all these metabolism which happens in the fasting state. Right. So here the hormone which is important in the fasting state is what? Glucagon. Glucagon will be increased in the fasting state. So we'll start off with liver glycogenolysis. Glycogen gets broken down. Liver glycogen gets broken down to give us glucose, and the glucose can be used up by the brain. Yes, brain always tends to use only glucose, and it can also be used up by the RBCs, and where it can produce lactate, and lactate can be a precursor of gluconeogenesis, so it can produce glucose. Similarly, glucose similarly, adipose tissue, the mobilization of triglycerol, the breakdown of it gives us fatty acids and glycerol. Fatty acid can undergo oxidation, fatty acid oxidation in various organs, right? Fatty acids can undergo oxidation in various organs like liver and skeletal muscle. Yes, and give acetyl-CoA. Now, excess of acetyl-CoA, if it cannot be if it cannot enter into TCA cycle, it will enter into the formation of ketone bodies. Yes, in the liver. So liver produces ketone bodies. These ketone bodies can be used up by the skeletal muscle. All right. Now the glycerol which is being formed is also a gluconeogenic precursor. So it will help in the synthesis of glucose again. Right? And then proteins, muscle proteolysis at the end stage, prolonged starvation gives us amino acids. These glucogenic amino acids can also be utilized for the gluconeogenesis pathway, right? And even kidneys can help in gluconeogenesis. Yes, as a long-term fasting, even kidneys can help in gluconeogenesis. It forms the alpha ketoglutarate, which is an intermediate of TCA cycle, which can enter into the gluconeogenesis pathway. All right, so this is the integration of all the metabolic pathways that can happen during the fasting state. Do you understand this? Yes. Thank you all.